Oh, do you want me to turn up my mic?
Yes.
Okay, and the video too, or?
If you'd like, I mean, if you'll be putting on the screen, your hand, it could help to.
Okay, so
Sure.
let's see. Like this?
Yep.
Okay. We have? I don't know.
We have 6 faculty members. I think we have 6 students.
So Xiao Chen is not here. Is he? I presume he's coming. A boy. Do you have any students? Maya must have students, right?
Yeah, my family is a student. Originally from Pakistan or Iran? Pakistan. I wish we would encourage more students to come to our office.
Well, we should encourage students to come to all seminars. But yes, abstract algebra not interested. Okay. Well, if you attended the colloquium on Monday, you would have probably changed your opinion. I mean, it was really fascinating stuff, using a lot of abstract algebra constructs. Fairly complicated combinatorics. Oh, speaking of the colloquia, next Monday, so we have a biomath colloquium, the biomath speaker, and I know the guy since he was a grad student, a long time, and Levin also knows him, right?
Whereas we actually had some overlap in the academic from the same group.
Okay. So he will give a talk in the colloquium on Monday, and also he will give a talk in computational medicine group on Tuesday. Yeah, Reinhardt's group, yes, and it will be a different talk. So at least I think it's at 4 o'clock in the
Wait,
it's in Shan.
it's Tuesday.
It's Tuesday, yes.
Yeah, the computational medicine they have the seminar usually on Tuesday at 4 o'clock.
That's right. So that he's going to give a talk in that time slot. I don't know, they will send us the announcement, but
Yeah, I can forward the announcement.
All right, let's get started. So it's my pleasure to introduce the man, myth, and legend, our very own Sergei Pilyugin. It's very nice to have you give a talk. Sergei received his PhD in mathematics, excuse me, at Emory under his advisor, Professor Waltman. And he's been a professor at University of Florida since 2000, yeah, since 1999.
yes, it started in 99. It's hard to believe. Yeah, but it's been like what, 26, the end of my 25th year.
And so he's been a full professor since 2009 and has published dozens of papers in population dynamics, stochastic dynamics, applying dynamical systems methods to biological problems. So please take it away.
Okay, thank you very much. I feel humbled by this introduction.
You established the biomass group in our department.
I don't know if it's really fair to say that, because we had Professor Kiesling, who was also, he was originally a biologist who later turned up, so he started, becoming more and more interested in modeling. And so there was a big effort to understand the citrus greening, and he took part in that. And he also brought Bert Singer to UF, essentially, who's also a prominent mathematician, probabilist, and later turned sort of a biomath person as well. Anyway, it's great to have this opportunity, and thank you very much. This talk is a modification of a talk that I gave in biophysics group in the fall. So some of the slides are maybe intended more for people in physics, but I will talk a little bit about mathematics at the end. So what is microbial kinetics? Right, and how do I, there. So I will talk about applications, and the first example would be batch growth, which goes back to Jacques Monod and his experiments. And then I'll introduce what continuous cultures are and continuous microbial growth, and the device that was first concocted by Novik and Zillard, the chemostat. And then I'll talk about the models that we actually use to understand these devices, including mutations, including competition, and the results on competitive exclusion principle. And then finally, I will actually shift to what we are doing now, and this is modeling biofilms. And most recently, doing a model of bioclogging. So what are the applications of microbial kinetics? Any process where microbial growth is involved is part of microbial kinetics. For example, wastewater treatment and bioremediation, where you purify a contaminated soil. Do I need a? I'm okay. I can point that way. But thanks. I don't think I have anything to plug it into, or maybe I do. Okay, so also the digestive tract, okay, and intestinal microflora is abundant with bacterial cultures. And in fact, if something goes askew, then you start getting a disease of sorts. So it's important to understand that. Biofilm formation, it's important, particularly if you're dealing with some sort of pipes where they may become clogged and prevent proper functioning. Bacterial infections, okay, because bacteria are a primary example of a microbe, okay. And so treatment of bacterial infections with antibiotics is part of microbial kinetics. controlled production of microbial metabolites. So a lot of chemicals are synthesized as byproducts of metabolism, including antibiotics themselves. And then finally, food fermentation. So this is ubiquitous in food industry, where you use microbial processes to make things ferment milk, so making cheese and things like that. Of course, fermentation of sugars to produce alcohol, etc., etc. The cells that participate in the functions of the immune system are also behave in a way like bacteria, because they are microbial organisms with a different function, but nonetheless, some of the mechanisms are similar. So you could use microbial kinetics to understand functioning of the immune system as well. So let's go back to the beginning. Well, one of the pioneers of microbial kinetics was a French biologist, Jacques Monod. And he actually got a Nobel Prize in 1965 for a description of the lac operon, which is a mechanism in E. coli. So you're probably familiar with E. coli. It's one of the most common microbial organisms. Okay, so if you read about food poisoning, it's very likely that it was with E. coli. So anyway, Monod studied the E. coli extensively, okay, and came up with this functional form for the resource uptake, okay, of the form ms over a plus s. So this kind of function, although at the time it was sort of a purely empirical observation that a function like this describes the kinetics very well. And he published several papers that are seminal in actually understanding how the microbes interact with the environments in terms of, taking up resources, et cetera, et cetera. Monod also described the effect of dioxy, okay, which is a two-phase growth exhibited by E. coli, where you start the culture, okay, and you supply it with two growth resources, well, say glucose and something else, lactose maybe. And interestingly enough, the microbes show this preferential uptake. So they first consume glucose and they grow on that, and then there's the growth stops temporarily, right? And then they restart growing, but now they consume lactose and convert that into biomass. Okay, and it was termed dioxy for dual growth. And understanding that was also a big question in how that happens. And all of that later on turned out to be effect of various transport enzymes, okay, that allow the microbes to bring in the resource from the environment. Okay, so these are the growth rates. Basically, what I did was just copy and paste figures from original papers by Monod. Okay, and so this was 1949, where he grew E. coli in various mediums and observed the growth rates at different concentrations, and basically said that, well, this function fits well. And if you were to use equations to describe that, then you might want to use, say, a simple system of theories where you have S for the growth resource and X for the biomass. And there's a conversion of a resource to biomass with a certain coefficient, which is the yield, capital Y, all right? And here, capital F of S is the rate, the specific rate of uptake, okay, as a function of the resource. And then you can just analyze what happens in the system. And you realize that because of there's a fixed ratio at which the units of the resource are converted to biomass, you have a conservation. So the sum, the linear combination of the two remains constant. And so you can further reduce the dimension of this to a single ODE, and then you end up with something like this, all right, where you still have this growth function. And if the growth function were purely linear, then you would end up with a classical logistic equation of growth, okay, where you have a fixed carrying capacity, which is determined by how much resources available to the microbes, okay, and then no matter where you start, you will end up with all of the resource converted to biomass eventually. And then that's when the growth stops. Okay, so now enter the new phase of development, and this is due to two people, Aaron Novick and Leo Szilard, and they were two physicists from the University of Chicago. And if you were paying attention, okay, these guys were also part of the Manhattan Project, which was the development of atomic bomb in the United States. And when that was done, they wanted to find something else to do. And they said, you know, from now on, we'll study mutations and genetics. And so they switched to actually working with microorganisms, okay? And if you want to study mutations, then you want to maintain the same culture, okay, in the same medium for a very, very long time. And so they came up with this concept of a continuous culture, where as opposed to the batch where you just inoculate everything, okay, on a dish or maybe in a tube, okay, and then leave it be, and continuous culture is constantly supplied with fresh media. And so here is, in the picture, there's actually, so this is Leo Szilard, this is Aaron Novick, and this is the laboratory where they made up the first chemostat, okay, the first continuous culture device. And they gave a formal description. As you can see here, you have this culture medium where, oh, sorry, the growth tube is actually here. So this is where the culture is maintained, and then it's pumped into the main reactor, all right? And a constant volume is maintained here. This is where all the growth, microbial growth happens. There are a lot of other things involved here to maintain sort of a controlled environment where you have to make sure that the coming medium is sterile, so you don't pump in extra microorganisms, and so on and so forth. Okay, so the equations change if you are switching from a batch to the continuous culture, because now you have to count for the flow rates, okay? So capital F is now the flow rate, and V times S is the total amount of resource in the volume V. X times V is the total amount of the resource, of the microorganism or biomass in the vessel, okay? And then, assuming that V is constant, right, you can divide by it, and F over V will become what we'll later call the dilution rate. Okay, so the dilution rate, if you ever taught an ODE course, right, you probably explained how this dilution process works, and that the reciprocal of D, reciprocal of the dilution, actually represents the mean residence time, okay, of a single molecule in this well-mixed reactor. If you just allow it to dilute. Okay, so and if you analyze the two equations, then you will see that this model exhibits a fairly simple dichotomy, where depending on whether, so S naught is the concentration of the resource that is supplied, okay, through the feed tube. So if the growth rate at this resource concentration is less than the dilution, then the culture will be washed out, okay, in the long run. And if the growth rate is greater than D, then there's a certain positive equilibrium where the culture will stabilize, and the positive equilibrium maintains both the organism and the resource, okay? And there will be at certain concentrations.
So, yeah, it doesn't really depend on the choice of the function.
Well, this model is formulated for just a generic function. Typical assumptions are that this function little f starts at 0 and then it's just increasing, and then it may saturate or it doesn't have to saturate. It doesn't really affect the mathematics. Later, when people got bored with studying these kind of models, they started introducing some other effects like inhibition at high concentrations, where this function starts going up initially, and then after passing through a certain maximum, it starts declining, okay, for some reason, which, of course, you can make an argument for why it would happen. But anyway, so non-monotone growth functions were also considered. What happens is that it introduces an extra steady state, okay, which is typically dynamically unstable, so it won't be observed in the long run anyway, right? But it's what it is. Yes. So, but when you do the analysis and at the end of the day, you have to do some sort of simulations, right, on the computer, you have to choose which function you want to use, okay? And the typical choice is Michaelis-Menten or Monod function. Right. So essentially, if you look here, if you consider this curve and the plot of the input concentration versus the dilution on the y-axis, all right? And for a pair of the two parameters, okay, if you're below the curve, you'll end up with a washout or extinction. And if you're above the curve, then you will maintain the culture. The culture will persist. I will explain what formal definition of persistence is a little bit later. Okay, now this is a single, as far as a single culture is concerned. So we're assuming that all organisms are genetically identical to each other, okay? And of course, the goal of Zillard and Novik was to study mutations, okay? And so the idea is if you maintain your culture over a long term, all right? So over many generation times. Oh, and this is another thing that I want to explain. So even though your culture is maintained at equilibrium, so that is constant concentrations of both the organism and the resource, it doesn't mean that the organisms are just sitting there, okay? They're constantly turning over. And the constant concentration corresponds to the balance between growth and washout. Right, so that is why they thought it would be a great device to use to study mutations, because as time goes on, right, then with with a large number of divisions, okay, you will start seeing mutations, right? And so what will the mutation do? Right, so a mutation will introduce a different species, right, in some sense, okay, which will either be a better competitor or a worse competitor, okay? And then if it's the worst competitor, will be washed out. If it's a better competitor, you expect it to actually displace the original culture. And that's exactly what they observed in the experiments, that they started with one culture, they call it the standard strain of E. coli, and after, I think, 5 days, it was replaced, okay, by a different culture, which they call the fast E. coli. Was basically a mutant which was more adapted to that environment. It just displaced the original strain, the wild type. And mathematically, this corresponds to this idea of a competitive exclusion principle, all right, which says that if you are growing a number of different microbial strains, okay, and your growth medium contains a single growth limiting resource, right? So essentially, you have several different species competing for the same resource. Then what you should expect is the survival of the fittest, okay, which is the strain or the organism which is best adapted to this growth medium, okay, will survive and will drive every other competitor to extinction. So this is what the competitive exclusion principle is. And in terms of these equations, that basically means that you look at the level of the resource, which is called lambda I, corresponding to the single species equilibrium for each of those competitors, all right? And you see which one has the lowest, and this is called the break-even concentration. So which species has the lowest break-even concentration? That's the one that is the fittest, okay? And that's the one that will actually drive everybody to extinction, okay? And it was shown mathematically under some assumptions. For example, the result is known if the growth rates are monotype. Later was generalized to other types, but I think in general this question remains open. Like the very complete proof of the competitive exclusion principle still remains elusive to this day. Like, and by this I mean for general growth rates. Okay, so the first proof, okay, was given in 1978 by Professor Xu that Xiao Chen studied with in Taiwan before coming here. And so Youngmin mentioned Professor Waldman. So Xu was also a student of Waldman's. And so both him and Hal Smith were the very first students of Waldman, and I was the very last student of Waldman. So we did share the same advisor, but on a time span of 20 years, roughly. So anyway, Professor Xu came up with a Lyapunov function that made everything work, okay, and it was utilizing this algebraic identity about Monod functions that if you consider these differences and these ratios, then they'll always come out to be constant, okay, and that's why everything works. Okay. So later was a lot of work trying to generalize this these Lyapunov functions, and Professor Wolkowitz and her students were actually among those who made probably the most progress in that direction. So anyway, but this is assuming that you don't have mutations. This is only assuming that you got to the point where you have multiple competitors, right? And if no additional mutations occur, what you should expect is the competitive exclusion, where the fittest survives and everybody else is driven to extinction. But in the presence of mutations, typically, so you may consider, for example, a model where you have the original culture, X1, and the mutant, X2, which is produced roughly at a very slow rate, okay, each time the first species divides. All right, and then it can grow on its own, et cetera, et cetera. And then you can study the equilibria of this new model, okay, and you'll see that either, there are two possibilities really. So if this mutant is a better competitor, then it will immediately replace the original strain. But if it's a worse competitor, then it will be maintained at a low level just because there is a constant transfer, okay, from X1 to X2 by mutations. So, yeah. Okay. And basically this idea of a break-even concentration as the single quantity that determines who is the best competitor, you can think of it in terms of two growth curves for two cultures. If they're, say, given by this blue and the red curve, all right, then depending on where the so this along the axis, I still have the resource concentration here and the dilution rate here, all right? So for example, in this range, okay, if you maintain the dilution in this range, then so the red curve will be a better competitor because it has a lower growth break-even concentration. But if you switch the dilution here, then you see that the blue curve now becomes the better competitor, because the break-even concentration is the x coordinate of the intersection of this horizontal line with the corresponding growth curve, all right? And so now you see that if, for example, if you were to vary the dilution, okay, maybe between this value and this value, then part of the time, one species will have the advantage. The other part of the time, the other species will have the advantage. And if you do it in a balanced way, then maybe you can make them coexist. And this is sort of driving at the temporal effect. I mean, for example, temporal fluctuation, like a seasonal variation of things, right? So temperature or other things. If something varies periodically, it could induce coexistence.
So this is that plot saying that both populations exist, they're both non-zero, but one is just greater than the other. And then there's a particular.
No. What it's saying is that if you maintain the dilution in this range, okay, not in this range, at this particular value, then the red species will drive the blue to extinction.
Oh,
But if you tune it up to go to this level, okay, then it is reversed. So at different dilutions, then you have different species being best competitors. Yes.
Yeah, and that was just thrown off by the resident and mutant strains coexist label.
Yeah, this is sort of okay. I'm trying to going back. So essentially, I just skipped the story, the effect of mutation. You can, I think I may have said it, but essentially, if the mutant is a better competitor, we'll just displace the resident, right? But otherwise, it will be maintained at a low level. Okay. So, and then which one is better, you can determine from this graph, right? Okay. So the biodiversity, of course, is abundant in nature, and which mechanisms could facilitate coexistence. So the resource diversity, for example, right, where you have multiple competitors and each one has their own preferred resource to grow on. Okay, and that way you can maintain more than one species. Temporal heterogeneity, where you vary parameters, okay, of your environment in such a way that some part of the cycle favors one competitor and another part of the cycle favors another competitor, and on the balance, they can coexist. You also have spatial heterogeneity, which is something I have not mentioned yet, but for example, imagine that each of these competitors chooses their own temporal spatial niche where they congregate to, okay, and then one will have advantage in one part and the other one will have advantage in the other part, and overall, again, they are able to coexist. And then finally, you also have the physiological variation and adaptation, where depending on the conditions, they can adjust, right, to, let's say, adverse environment versus the not so adverse environment. And you also have additional modes of interaction, like symbiosis and stuff. Okay, so this is the part of the ecological considerations about microbial growth. And now what I'll talk about from now on is sort of closer to biofilms, understanding biofilms, etc. So far, in all of the previous models, the microorganisms were considered as basically single cells floating in some solution. All right. But when you start talking about the actual organisms, right, the actual bacteria, some of them can stick to each other, right? And so they start growing in clumps, all right? Or people call them floccules. So this is the flocculation model. Basically considering exactly what I said, that you may have two different stages. One, you have the single organisms, and you can have also aggregates. All right, and they will behave differently because, for instance, you might argue that larger aggregates may have a different residence time. Okay. And the ultimate will be if these aggregates actually attach themselves to the walls of the vessel or some other part that is not being washed out. Okay, and in that sense, we will end up with a biofilm wall growth model after this. So, but in these equations, what you have is you have U and V. Okay, so U is a single organism, V is the floccule or clumps. Okay, each one has their own rate, and each one is diluted, but the dilution rates may be different, maybe the same, could be different. Okay, and then you have switching. So this is the rate at which they aggregate, and this is the rate at which the clumps are actually broken again back into the single organisms. Okay, so French group studied this in a number of papers, and they actually showed that. And I want to say, sorry, this is fairly recent. I mean, this is like 2017. They probably started a little bit earlier, maybe 2010 or so, but it was still this millennium. And they showed that this flocculation model exhibits a fairly rich dynamics. Okay, depending on the parameter combination, you have the following possible outcomes. So you may have the washout to be globally attractive, where you lose the culture completely. You may have if the dilution rate is sufficiently low, then you have the washout is unstable, and so there's a globally attractive positive equilibrium where you have both single organisms and clumps coexisting stably. All right. If you keep increasing the dilution rate, you may have bistability where there are two positive equilibria, okay, and they're both locally stable. Oh, sorry. So E naught, the washout becomes stable, and there's a positive equilibrium which is also stable. And there's another one that separates the two, which is a saddle. So depending on the initial condition where you start, you may end up either with losing the culture, it goes to extinction, is washed out, or they may coexist. Okay. And then later, after a few papers, they actually discover an interesting fact that if the dilution rate is diminishingly small, like a limiting argument where it goes to zero, you may induce oscillations via Hopf bifurcation, right? And this is very recent. In fact, I just reviewed a paper last year where they keep developing this concept of Hopf bifurcation in the flocculation model.
Unobserved in experiments?
No, I don't think so. It's purely theoretical. In fact, oscillations were observed in microbial cultures, but not in the laboratory setting, but rather in soils. Right. So there was some papers from the Soviet Union where they studied soils and they observed some oscillations. At some point, I had to dig up some of the experimental evidence for oscillations because we also did a paper where we showed that if you move away from the assumption that the conversion, the yield is constant, okay, so the efficiency of biomass conversion is varied depending on the resource concentration. You can also induce oscillations, and you can actually induce very interesting and complicated dynamics. And that was a while ago. But I just remember that you get a whole bunch of bifurcation types. You know, you have a period doubling, you have a Nymark stacker, all of them, Hopf bifurcation. Just all in the fairly simple chemostat. And we wanted to see if there was some experimental evidence for that, right? And it does exist, yes.
So once they are aggregated, is there a chance for them to dissolve and become as egg again?
Yes, they are. Sorry, that's what I was trying to say. So this is where they,
Oh.
right, so they switch back, right? They just unclump or desegregate, yeah. Yeah.
So, and also they are used a different unit between A and B. Yes.
Sorry?
So the parameter A and B has the different units.
Yes, A and B have different units, correct, because here, well, they are assuming that the way you aggregate is you build on the existing aggregate, okay? Or you can just have two molecules come together. So this is U times U versus U times V, right? Where you just add a single molecule to the already existing aggregate or to another molecule like that, right? And when an aggregate dissolves, it all immediately dissolves into small molecules, which is a big assumption. But I'm sure that you can build a cascade of equations where you continuously build up the size of the aggregates, okay, and study what happens there. And I'm sure they actually did that. And they probably didn't find anything interesting, so that's why they didn't study it further. Right. Yes, so right, and this is the simplest possible model of that scenario where you just have single organisms and then flux. Okay, so this is what the flocculation model gave us, and now we come to what Xiaochen was doing, or is doing rather. So imagine now that the bacteria can not only clump to them, you know, they can not only aggregate, but they can also attach themselves to the walls of the vessel. So like the soaps come on the shower walls, right? That's an example of a biofilm. So similarly, what if they attach themselves to the walls of the reactor? The immediate consequence would be is that they become immobile, right? So as the culture, the contents, the fluid contents of the culture are being constantly diluted, okay, these guys that are attached themselves to the wall, they will be sort of impervious to that, all right? So they're just sitting on the wall and their residence time goes to infinity. So that way they will have an advantage if it comes to, say, competing against other things, right? And we're trying to understand what effect it has on the overall dynamics. And so in what follows, we will have, again, three variables, S, U, and W. So S will correspond to the concentration of the growth medium. U will be the bacteria in the fluid form, and W will be the amount of bacteria on the walls, so the attached ones. And now, right. So I studied this, a version of this model for my PhD thesis. So it was a while ago. And at that time, we simply neglected the volume of the biofilm. So if it doesn't affect the amount of fluid in the reactor, then the growth of the biofilm should not affect the dilution rate. So the dilution will remain constant regardless of how much biofilm growth you have. That was the assumption then. And so you end up with the equations of biofilm growth with constant dilution that have this form. So you have the resource, which is supplied, it is consumed by both fluid form and the wall growth. Okay, and then you have here, so the fluid bacteria are, they grow, they are diluted, okay, and then they can attach themselves to the wall, and then they can, so the wall can detach back and become fluid. So you have a back and forth, right? So you have attachment rate and detachment rate. And for simplification, okay, I already scaled away the growth yields. So the growth yields are always assumed to be constant and equal to 1. And it turned out that this model really is just a very fairly simple dynamic picture, and that is you only have 2 outcomes. Either you lose the entire culture, meaning you lose both the organisms in the fluid as well as on the wall, or they can persist, and in that case, the culture will be maintained at a positive equilibrium. Okay. So in each case, you have a unique, globally attractive equilibrium. But now, when Xiaocheng came here, we started looking at what happens if you no longer neglect the volume of the biofilm. And specifically, in this could become relevant in the so-called microfluidic devices, where the actual volume of the reactor is minute, is very small, right? And so if you have any biofilm growth, it could pause, it can block basically the flow of the medium through the reactor. Okay, and the simplest way to understand this is, well, imagine that by increasing the volume of the biofilm, right, you effectively decrease, right, the amount of volume which is which corresponds to the fluid, right, in the reactor. Okay, and so if you just basically write down this equation, so the total volume is the sum of the volume of the biofilm and the volume of the fluid. Okay, and then you can express the reduction. Oh, and the dilution rate is still the flow rate divided by the effective VF, right, which is this difference between V naught and VW. Okay, and then you can express the dilution as a function of the wall growth, W, right? And here, this W naught is the amount of biofilm that completely clogs everything. So this is the amount of biofilm that takes up the entire available volume of the reactor. And this is, of course, in the denominator of the dilution. So as W approaches this critical threshold, W naught, the dilution goes to infinity, right? Because you keep pumping things at a constant rate, okay, but the actual volume that you're dealing with is diminishing to 0. So the ratio goes to infinity. And that makes things completely different, right? So this new model, where you just replace the dilution by this function of W, is what we are studying now. Okay, so I took the previous model, and instead of D naught, I put a DW here and no other changes. Okay, now you can imagine that if something goes to infinity on the right-hand side, you have a singularity in your model, right? And this is the novelty of this model, that is, it is not well posed, unlike all the previous models. In all the previous models, you had automatically, all solutions were defined, non-negative bounded, and defined for all positive times, okay? Here, it is possible that for some solutions, some initial conditions, okay, and under some parameter combinations, you will have a solution that where W approaches W naught, okay, and then maybe you hit a singularity in finite time, and then you cannot further extend the solution for all possible positive times, right? So speaking in mathematical terms, that would correspond to the semi-flow, okay, or the solution operator to be non-complete, right? So flow is complete if all solutions are defined on 0 to plus infinity. So here, we don't know. In fact, we do, but it came out as a result of the analysis that there are solutions under some conditions that will hit the clogged state in finite time, okay? But to facilitate further analysis, we wanted to avoid dealing with the singularity. So we decided to switch to the rescaled system. And so the rescaled system is basically so you introduce a new time variable, okay? So you rescale the time, and that effectively multiplies the entire vector field here by this quantity 1 minus W over W naught.
Really cool, so it's like.
So, but first of all, sorry, we all know from the general course, right, that if you took a vector field and multiplied by scalar function, which is positive, you do not change the orbits. So the solution curves remain the same geometrically, right? You just reparameterize how fast the points evolve on those curves, okay? So you do not change, for example, things like stability of equilibria, right, et cetera, so the phase portraits will remain the same. But the advantage of that is now this new system, which we call the rescale system, is well posed, okay? And the solutions exist on 0 to plus infinity. And so you can employ this entire machinery of, limit sets and et cetera, et cetera, et cetera. Yes?
Oh, it's just very cool. It's like a blow up in time.
Exactly. Yes, you do a blow up in time. That's absolutely what it is, right? So from now on, we will talk about this rescaled system. And first of all, let's look at what happens if you just look at this limiting case. So if you plug in W equal to W naught, then this entire thing just goes away. Okay, and so what you end up with is a system where simple equations, S dot is 1 minus S, U dot is minus U, W dot is 0. First of all, W dot is 0 indicates that this entire plane, what is it? W equal to W naught is an invariant set. Okay. And it's also clear that for this system, U goes to 0, S goes to 1. All right. So all solutions converge to we call this the clogged state, which is the equilibrium of the rescaled system. All right. And it is globally attractive in that plane. All right. And then further, if you look at the Jacobian, the rescaled system, then you see that it has this upper triangular structure, 2 eigenvalues, always equal to -1. And the third one is equal to this quantity, okay, which may actually determine, it does determine, this is the only one that determines the stability. So EW may become unstable. And if it is, it will be a saddle with 2-dimensional stable manifold and 1-dimensional unstable manifold. Or it could be locally attractive, in case all 3 are negative, right? But when it is unstable, then it's also important that its stable manifold will coincide with this. All right. So this plane is always part of the stable manifold of this point. Now imagine that this quantity G1 minus beta is actually positive, so that the third eigenvalue is also negative. All right. And this clogged state is actually locally attractive. Then the solutions will converge to it exponentially fast. And so you can basically solve for how fast the rescale time goes to infinity, right? And then convert it back to the original time, okay? And show that as your solution of the rescaled system approaches EW, the original solution will also approach EW, but it will hit it in finite time. So this basically means that when the clogged state is locally stable in the rescaled system, in the original system, right, you will have some solutions that will hit the clogging or clogged state in finite time. Okay, this is again something that has not been a feature of any of the chemostat models that I know of. All right, where you have the singular state that you hit in finite time. Okay, but otherwise, let me give you. I think we're coming to this to the end of the talk. But so let me just give you the summary of the mathematical results that we discovered. So first of all, we showed that if the washout equilibrium is locally attractive, then it's globally attractive. Okay. Secondly, if the clogged state is locally attractive, it attracts nearby solutions in finite time. But we also proved that if beta is sufficiently small, then EW is actually globally attractive. That is, if you start with any positive solution, you will hit the clogged state in finite time. In the original system, in the rescaled system, of course, time goes to infinity. It is also possible that both extinction and the clogged state are unstable, in which case they're both saddles. All right, and then then the model is uniformly strongly persistent, and there exists at least one positive equilibrium where all of these coexist. Okay, and we also proved that any positive equilibrium is either locally stable or it's a saddle with one-dimensional stable manifold. There's no other possibilities. Looking at the Jacobian structure, for instance, you cannot have hop bifurcations, meaning that whatever happens, you will not expect the oscillations. So you will not expect oscillatory behavior. Although multi-stable scenarios are feasible. That is, you may have multiple equilibria that are stable simultaneously. Okay, so and we discovered two scenarios where EW and some positive equilibrium are stable, and then where you have at least two positive equilibria that are both stable, and there's a third one which is a saddle. So you have multi-stable scenarios. And this should be probably the end of the talk that I gave in physics. And then, of course, I decided to throw in some mathematics, but we don't have the time. So I'll just show you these are simulations. Okay, so numerical examples. This first one is where the clogged state is globally attractive. So there are no positive equilibria, and the washout equilibrium is actually where U and W are equal to 0 and S is equal to 1. So it's right there. Okay. This W, so W equal to W naught is the plane that goes like this. All right. And so the simulation shows that if you choose some random points, okay, in this cube, right, then solutions converge to EW. Here, second example is where you have bistability. So the clogged state, EW and E sub C, these are the two stable equilibria, each one with its own basin of attraction. And then there's an EC1, which is a saddle point with that basically separates the two basins. The stable manifold of EC1 separates the two basins of attraction. And then finally, third example is where EW is now unstable, but you have two stable equilibria, EC1 and EC2, and you have an EC3, which is a separatrix, okay, between the two. So that's probably all I wanted to say about this. So just a historic note, which I already mentioned, that this work, the recent work, is part of Xiaochen's thesis, and the analysis of the constant dilution was part of my own thesis about 30 years ago. So the continuation of things. Thank you.
For the mark equilibrium stability, is it possible to, determine the region of this attraction? For example, given in this region, it will convert to this equilibrium, another region it will convert to another equilibrium.
I don't think there is an analytical way to do that, because what you're basically asking is, can we analytically describe the stable manifold of the separatrix, which is a very difficult task. So I would probably say no. You can determine the tangency of the stable manifold at the corresponding saddle point simply by looking at the eigenvectors corresponding to negative eigenvalues and looking at their span, right? But that's just a tangency. To extend it further out from the saddle point, you probably need to do numerics, right? So that's a question for you.
And here
You should, yeah, when you do the orals, somebody might ask you that,
Because the example will be.
Okay, perfect. So he'll ask you that question.
Or if you only have like a plan, then you can maybe run some simulation to determine the region of the attraction. And here you have the three.
It's,
three variables.
yeah, it is three-dimensional, right?
Seems like this is about the projection of the face portrait.
I would be careful with projecting because you don't know which way it sort of curves, right? If you project the wrong way, it may fold onto itself.
Yes.
So we don't know that. We don't really know anything specific about that stable manifold, right?
So we should wrap up here.
Okay, sorry.
Yeah, no
Yeah,
problem.
thank you.
Thank you very much.
Right.